Okay, Assalamualaikum and a very good morning, everyone. I am really, first of all, I would like to apologize because I am down with flu, so I can't conduct the class today by Zoom, so I have to record it. So we will continue our lecture today on topic 10, virus, bacteria, and archaea. Okay, we will still be on chapter 20 from the method textbook. So you can refer it from page 361 up until page 370. Okay, for the outline, okay, we've covered 20.1 last time, and then today we will be covering prokaryotes, bacteria, and also the archaea. Okay, so the first subtopic will be on the prokaryotes. So your learning outcome will be, so by the end of this lecture, you'll be able to describe the evolution of prokaryotes, identify the structural features of prokaryote, and also describe at least 4 ways in which cells of prokaryote differ from the eukaryotic cells. Okay, so the prokaryotes, we've briefed through a bit on prokaryotes in chapter 3, if I'm not mistaken. So let's recap on what is prokaryote. They are including bacteria and archaea, which are fully functioning living single-cell organisms. So when I say prokaryotes, it means before a nucleus, prokaryote. Okay, they are macroscopic, and they are about 1 to 10 micrometer in length and 0.7 to 1.5 micrometer in weight. They are abundant in air, water, and soil on most objects. Okay. It was first discovered by the microscopist, he's a Dutch, called Anthony, in the 17th century. So after that, a lot of scientists carried out various experiments to determine the origin of the microorganisms in lab cultures. So finally, in about 1850, Louis Pasteur, this guy, he devised an experiment for the French Academy of Science, which we'll see in the next slide. It showed that a previously sterilized broth cannot become cloudy with organisms' growth unless it is exposed directly to the air where bacteria are abundant. Okay. Okay, so this is his experiment. So he had two hypotheses. Hypothesis A is bacteria arise spontaneously in the broth, and hypothesis B is bacteria in the air that contaminates the broth. Okay, so in the first experiment, he boiled the broth. So he boiled the broth, right? And he placed this flask outside the building in open air, and he opened the lid up briefly. And it showed there is an 89% of bacterial growth inside that. Okay, so for the second one, he boiled the broth again, and then he opened it inside the building. Okay, and it shows 32% of growth. So what can he conclude from this experiment is that the hypothesis B is supported because the relative concentration of bacteria in the air explain the result. So bacteria from the air outside the building shows more growth, 89%. So he was to prove this, whether he has got the theory right. So he did the second experiment. He boiled the broth again, but this time he used like the thin narrow pipe or tube actually, and the flask is open to air at only this part here. So this means that the air here is pure. The air enters here and the bacteria collects here. So the bacteria does not go through to the flask. So there is no growth at all. 100% of no growth. So that means it comes to conclusion that hypothesis B is supported because when air reaching the broth contains no bacteria, the flask remains free of growth. Okay, so then Pasteur disproved the theory of spontaneous generation of microbes by performing this type of experiment. So he found that it is not a spontaneous thing. It has to be contaminated. Okay, so it is a non-spontaneous generation. Okay, so let's have a look at the structure of the prokaryote. Okay, so they lack a membrane-bound nucleus. Their DNA is in a nuclear region. They have outer cell wall. They move by the means of flagella. They lack membranous organelles. And they have the accessory ring of a DNA, which are called plasmid. Okay, so what they have are usually divided into three parts, which are the cell envelope, cytoplasm, and the appendages. So cell envelope, they have the glycocalyx, they have the cell wall, and they have the plasma membrane. While in the cytoplasm, they have the nucleus, the ribosome, and also some bacteria, such as the cyanobacteria, they have the thylakoids for photosynthesis. Okay, so appendages, they have flagella, they have pillars, and they have the hair-like fibrae. Okay, so this is the structure, the features of the prokaryotic cell. So firstly, they have the capsule, this thing here, the whole thing, okay. They have the capsule, which is the gel-like coating outside the cell wall, which are made up of a polysaccharide layer called the glycocalyx. And then they have the plasma membrane, okay, this part here, the red one. Okay, sheet that surrounds the cytoplasm and regulates the entrance and exit of molecule. And then they have the cell wall, okay, structure that provides the support and the shape of the cell. They have this hair-like thing, which is called the fibrae. They are hair-like bristles that allow the adhesion to surfaces. And they have the nucleus. Yeah, this is the location of the bacterial chromosome. They have this part here, which is called the conjugation pillars. They are elongated and hollow appendages used to transfer DNA to other cells. This part here are called the flagella. So each flagella of a bacteria contains the basal body. Okay, this part here. So this part here is the close-up. Okay. This is the basal body. They have a hook. Okay, hook that can spin. Okay, and the filament here. Okay, this is the filament. Okay, the red dash arrow indicates that the hook and filament rotate 360 degrees. Okay, like a ship, right? The fan, the ship. So it rotate 360 degrees for the motion to move. Okay, for the prokaryotic cell to move forward, they rotate 360 degrees. Okay, so this is the general feature of a prokaryotic cell. Okay, so reproduction in prokaryote, they are using asexual reproduction. Okay, so prokaryote reproduce asexually by means of binary fission. So this is called binary fission. So when conditions are favorable for growth, prokaryote divide to reproduce daughter cells. Okay, the same exact replicate. They have the same genetic material as the parent cell. Okay, so generation time is as short as 12 minutes. Mutations are generated rapidly and they are passed on to offspring more quickly than eukaryote. Okay, prokaryotes are haploid, thus mutations are immediately subjected to natural selection. So in eukaryotes, genetic recombination occurs as a result of sexual reproduction. So sexual reproduction does not occur in the prokaryote. But there are three methods of genetic recombination in prokaryote. They have the conjugation, they have the transformation, and they have the transduction. Okay, so in conjugation pillars, which is this part here, remember this the part sticking out, form between the two cells, which is the donor cell and the recipient cell. So the donor cell passes its DNA to the recipient cell through the pillars. Okay, they have like a conjugation bridge where the DNA is passed on to the recipient cell. So this is the duplicated plasmid. So in transformation, it occurs when bacteria picks up free pieces of DNA from other prokaryotes and becomes incorporated into the genome. So this part here, the genes are transferred from the donor to the recipient cell when the bacteria picks up free pieces of DNA. Okay, transformation. So in transduction, it occurs when bacteriophage carries portions of bacterial DNA from one cell to another. They are then served as vectors, so they need the help of bacteriophage in transduction. Okay, next we go on into subtopic 20.3, which is the bacteria. So bacteria are more common type of prokaryote. Okay, so we can't see them with our eyes, but the biomass of bacteria on the earth exceeds that of the plants and the animals combined. So they are massive. They are everywhere. Okay, they are found in practically every kind of environment on earth. Okay. So most bacterial cells, they are protected by a cell wall composed of unique molecule called the peptidoglycan, which is a complex polysaccharide linked by amino acid. So there are two types of bacteria that have been distinguished. They are based on the cell wall actually, so they are called the gram-positive and the gram-negative. So gram-positive bacteria, they have a very thick peptidoglycan cell wall. So this gram-negative have a thin wall. So gram-negative bacteria have a second plasma membrane which blocks the antibiotic drugs and making infections difficult to treat. Okay, so when they die, when they are washed after staining, the gram-positive bacteria retain the dye and appear purple, while the gram-negative bacteria do not retain the dye because they don't have the thick peptidoglycan cell wall, so they appear as pink. Okay. Okay, so this is the experiment on how you can distinguish the bacteria. So first thing first, we have the gram-positive and the gram-negative. So is crystal violet is applied. So both of the cell walls affix the dye, so they become purple. Okay, so and then the gram iodine is applied. So the crystal violet iodine complex form inside the cell, so all are one color. They become crystal violet iodine complex. After that, they have to be washed. So they are washed with alcohol, and the alcohol for gram-positive dehydrates the thick PG layer, trapping the dye complex, so they become purple. While the gram-negative, alcohol has minimal effect on the thin peptidoglycan layer, so they become like a brownish color. After that is the staining process. They stain it with the safranine, which is a red dye, and what happened? In gram-positive, they maintain the purple color. This dark purple masks the red dye. And the gram-negative, they become like a pinkish color or red dye, or a red or pinkish usually. In normal lab result, we will usually get pinkish color, not too much of a red. So the red dye stays the colorless cell. So from this color, it will become either a red or pink, like you see here. There are some purple down here, right? There are some purple and there are some pink. So there are negative and positive bacteria. So the thick peptidoglycan layer, you can see the gram-positive traps the crystal violet dye, so bacteria appears purple after the stain. While the gram-negative bacteria have much less Pg, so they do not retain the crystal violet dye and so exhibit the red counter stain. Okay. So other than the cell wall type, bacteria can also be further classified in terms of basic shape, which is the spiral or spirally. Okay, so this is where it gets the name spirilla. Okay, rod bacilli, which is bacillus shape, bacilli. Round, cocky or coccus type. Okay, so this is the coccus type, the roundish. Okay, on top of that, on top of the cell wall type and also the shape, bacteria can also be characterized by their growth arrangement. So for example, they have the staff arrangement, which is described the cluster of cell. So these usually are the staphylococcus staff. Okay, and the next one is strap. Strap arrangement describes chain, which is streptobacillus. Okay, strap, like a chain. This is here. So this is round and it is a shape like, that's why it is streptococcus. Okay, strap. For the last one is diplo. Diplo arrangement describes pairs, which is like a diplococcus type. Okay. So next we go on into the bacterial metabolism. So they need, some of them need oxygen and some do not. Okay, so that is the oxygen requirement. So the first one, they are obligate aerobes, which is they cannot live without oxygen. They need oxygen to grow. So the next one is obligate anaerobe. Okay, they don't need, so unable to grow in the presence of free oxygen. So they do not need the presence of free oxygen. Another one, facultative anaerobic. They are able to grow in either the presence or the absence of oxygen. Okay, presence or absence of free oxygen. So next we go on into autotrophic bacteria. So the first one is photoautotroph. What does it mean by photo? Photo are lights. Auto is self. Okay, this is light and this is self, and this troph means food. Okay, so they use solar energy to reduce carbon dioxide to organic compounds. They are photosynthetic. Okay, and there are two types of photoautotroph bacteria, which is the anoxygenic photosynthesis and oxygenic photosynthesis. So anoxygenic photosynthesis does not produce oxygen as their product, while oxygenic produces O2. So anoxygenic, they photosynthesize in the presence of oxygen, and they do not emit oxygen. They need oxygen and they also do not emit oxygen. So they use the photosystem 1 only. Example of bacteria are usually the green sulfur bacteria and some purple bacteria. They carry out anoxygenic photosynthesis because they live in oxygen poor condition. So in oxygenic, they use the photosystem 1 and 2. So an example, usually we use the cyanobacteria, because cyanobacteria contains chlorophyll A and they carry on oxygenic photosynthesis just as algae and also plants, and they give off oxygen as the byproduct. Okay. So next we have the bacteria called the chemoautotroph. So what does chemo means? Chemo means they rely on or they pertain to chemicals. Auto is self and troph is food. So they carry out chemosynthesis. Okay, they are chemosynthetic. They carry out chemosynthesis. So they oxidize inorganic compounds such as hydrogen gas, hydrogen sulfide, and ammonia to obtain the necessary energy to reduce carbon dioxide to an organic compound. So they usually live in environments such as deep sea vents about 2.5 kilometers below the sea level. Okay, so that was autotrophic. Now we go on into heterotrophic. Heterotrophic means they cannot produce their own food, so they have to depend on others. So most prokaryotes are chemoheterotroph that take in organic nutrients. So chemoheterotrophic bacteria called saprotroph, okay, they serve as decomposers that break down organic matter from dead organisms. So they are essential components of a healthy ecosystem. So these are chemoheterotrophs. They may be free living or symbiotic. Okay, so the first one we have commensalism, which is one population modifies the environment in such a way that a second population benefit from it. Okay, for example, obligated anaerobes live in our intestine because of the E. coli uses up the oxygen. Okay, so the second one benefits from it. Okay, second one is mutualism. Mutual, so both species benefit from the association. For example, we have one living in our human intestine is the release of vitamin K and B12, which helps to produce the blood components. Okay, both benefit from association. So the last one, we have parasitism. Parasite benefit at host expense. Okay, at the host expense only. So they are disease-causing bacteria. They are called pathogens. So and many of these form the endospore. Okay, so parasitism, you have to remember that only one species benefit, while the other one is harming the other. While in commensalism, only one species benefit, whereas the other is unaffected. In mutualism, both species benefit from the association. Okay, so the first one, the nodules of the Jew. Okay, so this is an example of the mutualism. Okay, there are some free-living bacteria carries on the nitrogen fixation. However, the bacteria of the genus Rhizobium invade the roots of the legume with the resultant formation of the nodule. So invade the roots and they become nodule. So nodules are formed, and here the bacteria converts the atmospheric nitrogen to be organic nitrogen so the plants can use it. So it is mutual. Okay, they get both benefit. So for this one down here, we have the endospore of the Clostridium tetani. Okay, remember when you were little, you have to do the tetanus shot, okay, injection. So this because when someone steps on a rusty nail, this bacteria can be injected deep into the damaged tissue and produces a toxin that causes the disease tetanus. Okay, so the bacteria, it produces a terminal endospore that causes it to have the drumstick appearances. So if endospores gain access to a wound, they germinate and release the bacteria that produces a neurotoxin. So the patient then develops tetanus and progressive rigidity that can result in death. Okay, and also this is what I meant by immunization can prevent tetanus. I think all of you must have gotten this tetanus jab, I think during Form 3. Okay, so parasitic bacteria cause diseases and therefore are called pathogens. So these are a few of the bacterial diseases in human. We have the sexually transmitted disease, which are syphilis, chlamydia. Okay? We have also this respiratory disease, such as the strep throat, pneumonia, whooping cough, also TB. Okay? We have the skin disease, which is the boils, okay, acne, infections, or surgical or accidental wounds, burns, okay, and also the Hansen disease. So bacteria that are disease in human for digestive tracts are usually caused by gastroenteritis, food poisoning, ulcers, and also the dental caries. Okay, dental goes down to our digestive tract. Okay. Nervous system, we have the botulism, we have tetanus, we have meningitis. Those are caused all by bacteria. And we have systemic disease, which are diphtheria, typhoid, and plague. Other diseases are tularemia and Lyme. Okay, we don't usually get this in Malaysia. Okay, so a number of antibiotic compounds are active against bacteria and are also widely prescribed. So there are two classes of antibacterial compound. So those that inhibit the protein biosynthesis and those that inhibit the cell wall biosynthesis. So these are examples of them for protein synthesis, erythromycin and tetracycline. While for inhibiting the cell wall biosynthesis, they generally block the formation of peptidoglycan, so which is a compound necessary to maintain the integrity of the bacterial cell wall. So these bacteria, they affect bacterial, but they do not affect the animal cell. So these are usually prescribed penicillin, ampicillin, and fluoroquinolone. Okay, whenever we are sick, usually from young, doctors usually prescribe antibiotics, right? So the more your body take in antibiotic, the more it becomes bacteria resistant. So it can be good, but however your body lama-lama dah jadi too immune to it, so it might not work anymore. Okay, that's why bila you dah makin tua, makin increase in age, bila doktor prescribe a specific antibiotic, maybe the same one that you used last time, it maybe does not work for you anymore. So you need a newer antibiotic, a newer type of antibiotic, a different strain of bacteria. So this is what's happening, and it's quite alarming to the world right now, okay? So genes are conferring resistance. They can be transferred among bacteria by transformation, conjugation, or transduction. And now, the 90% of the Staphylococcus aureus are resistant to penicillin already, okay? They become resistant. So we need a stronger antibiotic, okay? And they are increasingly to methicillin, which is MRSA. So MRSA is common usually in hospital and also nursing facilities. So what they are doing right now is they are trying to produce more stronger antibiotics that can treat most of the bacterial disease or bacterial infection. Last time when it started, when penicillin was first introduced, it was less than 3% of Staphylococcus aureus strain were resistant. Now there is about 90% of it. Okay, so scientists have got to be fast in producing the antibiotics. Next, we go on into cyanobacteria. So formerly, the cyanobacteria were called blue-green algae, and they were classified with eukaryotic algae, but now they are classified as a prokaryote. So cyanobacteria can have other pigments that mask the color of chlorophyll, so that they appear red, yellow, brown, or black rather than only blue-green. So cyanobacterial cells are rather large. They are about 1 to 50 micrometer in width. They are single cell. They can be colonial or they can be filamentous. Okay. They are gram-negative bacteria that are photosynthetic. They are believed to be responsible for introducing oxygen into the primitive atmosphere. They lack visible means of locomotion. They don't locomatize. They can live in extreme environments, but they are usually more common in fresh and marine waters, in soil and on moist surface. But sometimes they can also be found in harsh habitats such as the hot spring. They are mostly associated with fungi. They can form lichens, and they possess heterocysts, which are cells without nuclei, where nitrogen fixation occurs. Okay, this cyanobacteria can also result in the cyanobacterial bloom, which are the result from pollution in lakes and ponds. Usually, I'm not sure if maybe for those around Sri Kembangan, you have seen that some ponds, especially the one in Sri Serdang, it becomes brownish sometimes. It's because of the pollution and because of the bacteria, cyanobacteria. Okay, so these are the diversity about the cyanobacteria. Okay, they can be colonial. Okay, they can be colonial, which are single cells are grouped in common gelatinous sheath. And they can be filamentous. Okay, panjang-panjang macam ni. And then they can be one cell as well. Okay, there are 3 different diversities among the cyanobacteria. Okay, so the last one we'll go into is subtopic 20.4, which is the archaea. So the archaea were formerly considered as bacteria. So this archaea came to be viewed as a distinct domain of organism in 1977 when Carl Schwartz and George Fox discovered that the base sequence of the rRNA, which is ribosomal RNA, differs from the bacteria. Another difference they found out was that archaea do not have the PG, which is the peptidoglycan in their cell walls, like the bacteria. And also archaea are biochemically more like eukarya than bacteria, and is now thought to be more closely related to eukarya than to bacteria. So as discussed in section before, so it is proposed that the tree of life contains 3 domains, which is archaea, bacteria, and eukarya. because archaea and some bacteria are found in extreme environments, so they may have diverged from a common ancestor relatively soon after life began. Okay. Okay, so this side, okay, just never mind this. This is typo. I'm not sure why is it here. I must have copied the slide template. Okay, all right. So the archaea, there are three types of archaea, but all of them are originally discovered in living in extreme environmental condition. Three types are the methanogen, halophiles, and also the thermoacidophiles. Okay, so the halophiles first. The halophiles require high salt concentration for growth. Okay, they are usually found in the Great Salt Lake, okay, such as the Dead Sea, the solar salt pond, and also the hypersaline soil. Okay, salty. For methanogens, they produce methane from hydrogen gas and carbon dioxide. They are usually found in swamps, okay, in swamps area. While for the thermoacidophiles, they are the third major type of archaea. They usually thrive in extremely hot acidic environment, such as hot spring, geysers, submersible vents, and also volcanoes. They reduce sulfides and survive best at temperatures above 80 degrees Celsius. Okay, so the plasma membranes contain unusual lipids that confer tolerance of high temperatures. Okay, there are three types, which are anaerobic marshes, which are the methanogens, salty lakes, halophiles, and the thermoacidophiles live in the hot sulfur springs. Okay, so that is all for our chapter 20, our topic 10, sorry, viruses, bacteria, and archaea. So that's it for our lecture today as well. I think I have done the connect practice as well. You can do that after this. Yeah, you can work on your connect. Okay, I'm really sorry that I can't do the Zoom today because I am really unwell. I'm planning to go back soon. Okay, so always wash your hands thoroughly with clean water and soap during this pandemic. Okay, stay safe everybody. Assalamualaikum. Bye bye.